For this session, just want to say thank you so much for everybody being here. My name is Lisa Bilodeau. I go by she, her. I am Vice President of the Board on Lupus Ontario. I am also a lupus warrior. I have SLE and 3 other autoimmune conditions. And I really enjoy being able to volunteer my time with this organization. I learned so many things and meet amazing people, such as the doctors who have presented. If you are just joining for this session, please go ahead and look for the other recordings. We have 4 sessions for our Lupus Ontario Symposium this year with a theme of redefining the lupus landscape. And so we heard yesterday from Dr. Laura Whittle Garcia on Euler guideline revisions. We heard from Dr. Murray Urowitz about lupus types explained. We just heard from Dr. Amanda Steinman on biologics explained, and now we're going to move into our next session with Dr. Mark Matsos on CAR T-cell therapy and upcoming treatments. Dr. Mark Matsos is an associate professor in the Department of Medicine, Division of Rheumatology, McMaster University, and Rheumatology Head of Service at Hamilton Health Sciences. His academics and clinical interests include the management of systemic lupus, inflammatory arthritis, and young adults with pediatric onset rheumatic disease. He is a clinician with the McMaster University Lupus Clinic, investigator in multiple clinical trials, and medical advisor to Lupus Canada. He is a member of the Lupus Clinical Investigator Network, a collaborative organization of academic lupus clinics throughout North America dedicated to the identification and study of novel therapeutics to treat SLE. Dr. Matsos also works in close collaboration with pediatric colleagues in rheumatology at McMaster's Children's Hospital, working to improve the care of patients in JIA and childhood onset SLE as they transition to adult care. In 2018, he led the creation of the Young Adult Rheumatic Disease Clinic at McMaster's Forest Clinic in order to offer dedicated care to young adults living with chronic rheumatologic disease. Thank you, Dr. Matsos, for being here. And once again, thank Roche, our presenting sponsor, for supporting this symposium overall. In this session in particular, Dr. Mark Matsos will provide an overview of emerging treatments for lupus with a focus on the CAR T-cell therapy. He will explain the fundamentals of CAR T-cell therapy and discuss how this innovative approach could revolutionize the treatment landscape for lupus patients. This session will offer a glimpse into the future of lupus care, highlighting the potential benefits and transformative impact of these new therapies. And I'll turn it over to you, Mark.
Thanks, Lisa, so much for that kind introduction. And I want to thank Lupus Ontario again for the opportunity to pull out my crystal ball in a sense tonight and offer a glimpse into what we hope is the not too far distant future in terms of the management of lupus. I'd like to thank everyone for joining us today. And thanks again to Dr. Steinman for her excellent talk leading into this. So just some brief objectives here to start. I'm going to do something audacious and try to very briefly review 60 years of lupus therapeutics in about 2 slides and 2.5 minutes, just to set the stage of how we got here in terms of where we're at in 2024 and as we look ahead to the next generation of lupus therapies. I'm going to initially discuss those therapies that are in the near future. And what I mean by that are therapies that are currently in phase 3 clinical trials. And this is the stage of clinical trials that are really the make it or break it time for our therapies. And in order to come to market, like some of the medications that Dr. Steinman already talked to you about, like Benlysta and Safnello, any potential new therapies have to pass a very, rigorous set of trials that are run around the world with many patients to both prove their effectiveness compared to therapies that we already have available and to prove their safety as well, at least in the short term. So we're going to focus on those. Joe, I know you were interested in mTOR. I'm sorry, I'm not going to talk about that specifically tonight. And then what really the topic that has the entire not just lupus community, but I think rheumatologic world buzzing, is to chat a little bit about the hope around this CAR T cell therapy, and I'm going to leave you in a little bit of suspense until the end of my talk on that. Really briefly, here are some of the disclosures in terms of some of the relationships I have with industry and other knowledge translation organizations. Okay, so the name of my talk tonight was long time running, and you'll forgive me a little bit, I hope. When I was contemplating how I was going to approach this talk tonight earlier in October, it was around the release of the Tragically Hip documentary at TIFF, and of course last week the anniversary of the lead singer Gord Downey's passing, and I was a huge fan, so their discography was kind of top of mind for me, and when I thought about the journey that collectively lupus patients and their families and physicians have been on over the last 70 years, the long-term running sort of jumped to the forefront, and hopefully the second part of that tagline, it's well worth the wait. I'll prove to you as we go through this talk. So this is an attempt to summarize 70 years of lupus therapy in a single slide, and it goes all the way back to the 1950s, when in fact our anti-malarial therapies, meaning Plaquenil or hydroxychloroquine, that are still and remain our anchor therapies, as Dr. Steinman outlined in her talk, were first approved for the use of lupus along with glucocorticoids. And really our use of those two therapies in particular over the last 7 decades, while certainly imperfect, to some degree, did revolutionize, at least initially, the management of this disease, and side effects notwithstanding from the long-term use of glucocorticoids really proved critical in terms of our ongoing management, even to this day, of lupus. And what you see between the 1950s and really this century is a big kind of void of a lack of innovation and a lack of approved therapies, and even some of the medications that many of you have become potentially accustomed to being treated with, things like azathioprine or mycophenolate. Well, these are drugs that are used effectively in the treatment of lupus for many patients. They were never, in a sense, meant to treat lupus. These were not therapies that were designed to address the specific pathophysiology of the illness. And like many other areas of rheumatology, decades ago, our colleagues were kind of borrowing, and stealing from other therapeutic areas like oncology in an effort to treat the immune system and impart clinical benefit to our patients. And it wasn't really until the early 2000s that, in a sense, we developed a fundamental, basic understanding of some of the key pivotal targets in the diagnosis of, in the pathophysiology of lupus, and had agents that could target those, that we entered really what you can coin the biologic age, and a more, targeted ability to address these diseases. And so in and around 2010s, we had belimumab, which was the first lupus agent approved since the approval of the anti-malarials in the 1950s for the treatment of lupus. We went 60 years in between drugs. And then just a little over a decade later, we now have anifrolumab. And what I will say is we are all very optimistic that it's not going to be another 10 or 20 years before we see further treatment options added, hopefully, to this list, in an effort to do an even better job of addressing disease control. And I do say that as someone who's been fortunate enough to participate in some of the clinical trials of some of the agents that we're going to talk about. In particular, at McMaster, we were fortunate to participate in the anifrolumab trials before it was approved for therapy about 2.5 years ago. But the end result of all this drug development has led to this sort of playbook, which, again, Dr. Steinman sort of outlined for all of you in her talk. You can tell we read the same journals. Where drugs like belimumab and anifrolumab are now integrated into the rheumatologist's playbook when it comes to treating lupus, among some of our other more traditional therapies. Now, the challenge here is, as much as these therapies have been in the management of lupus, We know that one, unfortunately, not everybody's disease is going to respond to these biologics. We know there will be patients where, while they do initially respond, the response may not last. And we know, unfortunately, there's going to be some individuals out there who, although we get access and we attempt to utilize these therapies, they're just not able to tolerate it for any number of reasons. And so while they've been an excellent start in terms of our, ability to treat more specifically in lupus, we know that they can't be the only therapies that are ever developed because more options for many of our patients are ultimately going to be needed as we, treat increasing number of patients and we try and treat them over the course of a lifetime. And so the kind of begs the question in terms of what comes next. And so again, you'll recognize these photos, I think, from Dr. Steinman's talk, and really, highlight in black a lot of the potential therapeutic agents and targets that are currently in development, both targeting, both parts of the innate and the adaptive immune system. Now, we're obviously not going to cover all this ground tonight, not in the scope of our talk. But again, I'm going to do my best to address, some of the near available, hopefully, therapies. And despite all these targets, there's still two general themes when it comes to how we try to target this disease in a more specific way. And one of those is by targeting the type 1 interferon system, for which you already heard about anifrolumab tonight, and how we target the population of immune cells that are called B cells, which historically we've tried through drugs like belimumab and another drug called rituximab. So the first set of drugs that I'm going to chat briefly about are actually small molecules. And we talked a little bit about biologics and infusions and subcutaneous injections. But the small molecules are oral pills. And in fact, there may come a day when we have very effective, more targeted oral therapies to manage lupus. And these family of oral molecules in particular block signaling pathways within immune cells, something called the JAK-STAT pathways. And so what happens in cells is when cytokines like type 1 interferons are produced, they have to bind on a receptor on a cell surface. And once bound, they initiate a series of signals within the cell that lead to the production of further proteins and cytokines from the nucleus of that cell that help to perpetuate disease. And so one strategy, which is what anifrolumab leverages, is to use an antibody or a protein to block the receptor on the cell. So that although you may have a bunch of interferon present in the body and at high levels in lupus patients, it's not able to bind that receptor and lead to the downstream signaling that continues to perpetuate inflammation and disease in patients. But another way to target it might be to allow that binding, but inhibit a cell's ability to respond to it. And this is the category of the JAK inhibitors. And in particular, there's two types of JAKs that we're targeting in lupus. This is a family of 4 different proteins, and the ones most currently in development are looking at blocking JAK1 and TYK2, which I've indicated here on the arrows. And in particular, these are important to us because they're the JAKs that help to progress the signaling once type 1 interferons bind to the cell surface membrane. The advantages to a small molecule or an oral therapy over potentially a biologic are several. I will say patients, there can be a bit of trepidation in terms of moving to an IV or an injectable therapy. And by and large, I think patients do prefer pills, although I will say it also leads to difficulties in terms of compliance because it's hard to remember to take a pill every single day, as I'm sure some of you are well aware. With this class of drugs, we do actually have extensive experience in other rheumatologic illnesses. So this has been a strategy that's been deployed in illnesses like rheumatoid arthritis and psoriatic arthritis and psoriasis, and more recently now in vasculitis. So it's a family of agents and a strategy that many of my colleagues in rheumatology are well familiar with from having used them in some cases for more than a decade to treat some of our other illnesses. And the case of some of these other illnesses, they can be quite rapidly effective, sometimes showing benefits within the first 4 to 6 weeks of treatment. But on the other hand, there are some unique concerns that may become practically applicable, particularly to lupus patients. This has to do with the infection risk that comes from these therapies, in particular zoster, which is shingles. There is a potential cardiovascular signal with these medications, meaning that in some patient populations, but not all patient populations, we do see somewhat higher risks of coronary artery disease. And in some cases, we also see higher risks of clots. And so while they're an attractive therapy, they may not necessarily be the most appropriate therapy potentially in some of our lupus patient populations where these can already be significant risks. You see a number of potential agents here in the diagram, several of which are currently under investigation in lupus. And I'm going to give you a really brief overview in terms of where we're at in terms of the data behind the initial clinical trials of these medications. So the first of these medications I want to outline is called baricitinib, and this BRAVE trial was published last year as a phase 3 study. And this was a study that looked at patients with moderate to severe lupus who had positive antibodies and looked at the benefits of using this molecule over a year. And it was a medication that was compared with a placebo and in patients who are on background standard of care with drugs like prednisone and hydroxychloroquine. And when we look at lupus clinical trials, the way that we try and assess as to whether a drug is working or not compared to our standard therapy is by using a particular outcome measure, in this case, that's called the SRI-4. And what this looked for is this looked for a tool that we use in clinic called the SLEDAI to have an improvement of 4 points. And this is a standardized tool that's used by many rheumatologists day-to-day in clinic as a means of assessing how active somebody's lupus is. An improvement of 4 is felt to be, a significant, clinical improvement in that patient's disease. As an example, in a lupus patient who had active inflammatory arthritis, so lots of pain and swelling in the joints, an improvement of 4 points would identify someone whose arthritis had completely resolved. So an important marker. In this study, about 80% of patients were on steroids at the time that they entered into the study. And almost half of them were on doses of prednisone more than 10 milligrams. So these were patients with quite active disease, even though they were on, significant doses of steroids. And unfortunately, this study actually was not positive. And even though the strategy of blocking, in this case, JAK1 and JAK2, made a lot of sense from a pathophysiology standpoint, at the end of the clinical trial, we were unable to show a significant difference between the lupus patients who received the experimental therapy versus those patients who continued on with simple standard of care. And, I will say this was a bit of a dash, all of us in the lupus community, because we were certainly hopeful as the first, phase 3 study looking at JAK inhibitors that this was going to be something that was going to be positive and potentially, help to move things along. As with any time we have a clinical trial that unfortunately is not successful, we look at a lot of factors to try to understand why. In some cases, it was felt that potentially there was an issue with how steroids were handled in this clinical trial, meaning that patients weren't necessarily aggressively tapered on their prednisone dose. And sometimes that can lead to difficulties looking for improvements in the treatment group when the placebo group is on a drug that's still quite effective. And we also wondered if perhaps the SRI-4 was the right outcome measure to show us the difference in these trials. And there are other outcome measures out there that can be used that do a better job of looking for partial rather than full improvement of lupus patients' symptoms. So, this was an unfortunate first step into, looking at this class of drugs as a viable, therapy, but the good news is not the last. And that brings us to another medication that's in the JAK family, but targets a different type of JAK, something called TYK2, deucravacitinib, and this is currently in phase 3 clinical trials. So this is currently being studied. Patients are enrolled. We're hoping that within the next 12 months or so, enrollment completes so that we can get down to data analysis and have a good look to see whether this might be a better therapy when it comes to targeting the JAK-STAT pathways. What I've shown you here is data from the phase 2 trial of this medication. A phase 2 trial is an earlier stage of clinical development, almost more of a proof of concept before companies that are developing these medications choose to move on to much larger and more expensive clinical trials. If you're going to spend a lot of money running an international trial, you want to have some data that shows that at least at an earlier stage, you have a good proof of concept. And so phase 2 studies tend to be smaller and tend to be shorter, looking to see whether there's a worthwhile enough signal that it makes sense to move forward with your larger study. And so in this study that looked at lupus patients on this experimental medication over 32 weeks, fortunately, there was a statistically significant improvement across the board in terms of some of the key outcome measures that we look for that tell us that a therapy is potentially going to be useful. That included the SRI-4, which I've shown here. It included a BICLA response. BICLA is an outcome measure that allows us to look for partial improvement in patient symptoms. And it also included something called the Lupus Low Disease Activity Score. And this is an important new outcome measure for us in the lupus community because compared to some of our other tools, it's much more straightforward to use day-to-day in clinic. It, in fact, is part of the guidelines that were published last year by EULAR in terms of a target that we aim for in lupus patients. And we know that when lupus patients are able to achieve a lupus low disease activity score, we tend to see good things like less hospitalization, less mortality, less prednisone, less damage, and less flares of disease. And so you can see across the spectrum here of these outcome measures, this experimental therapy proved successful and why it was moved forward to a phase 3 clinical trial and why we hope that when that data is published, we do see some different results compared to that first study I showed you with the JAK inhibitor baricitinib. Now, there is in fact a third JAK inhibitor that's currently in clinical trial. And again, currently in phase 3 studies. So that last, hurdle before regulatory approval. And this is a medication called upadacitinib. And what I'm showing you here is, again, the results of a phase 2 study called the SLEEK trial. Upadacitinib, otherwise known as Rinvoq, similar to some of our other JAK inhibitors, is currently in use for several other rheumatologic illnesses. And so we do have a lot of collective experience with this therapy outside of the lupus paradigm. Again, in this phase 2 study, these are shorter, smaller studies, and this looked at outcomes at 24 weeks instead of what most clinical trials in phase 3 look at, which is 52 weeks. We once again saw across the board statistically significant improvement in patients who were treated with the experimental agent compared to those on placebo. Now, this trial also had a unique arm in which this JAK inhibitor therapy was used in combination with another immunosuppressive that targeted B cells more directly. But interestingly, there was not a significant improvement when you added a second therapy in. And so currently in the phase 3 clinical trial, upadacitinib, the JAK inhibitor alone, is being used to treat lupus patients. Also currently enrolling a little bit further behind the previous agent that I discussed, but we also hope and are eager to see the results of phase 3 trials and hopefully have, again, another useful therapy, an oral therapy to treat lupus. And so in summary, when we look at these small molecules and, the blocking of the interferon pathways through JAK-STAT inhibition rather than through anifrolumab, we have some mixed results at the beginning. We have negative results from our initial pivotal phase 3 trial of baricitinib, but we still have some promising results from other phase 2 studies. And really, the phase 3 results will be the pivotal data answering the question whether this is going to be an available pathway to treat lupus moving forward. And if shown to be successful, certainly long-term safety is going to be important for us to keep in mind, particularly with some of the safety data that we've seen in other illnesses outside of lupus, but which lupus patients are at risk for, in particular those cardiovascular and clot risks. So stay tuned. These agents may soon be coming to a rheumatologist's office near you, and may in fact, be a therapeutic option available for you to discuss with your rheumatologist. So moving to the other side of the equation, so you'll remember besides the interferon pathway, the other target over the last 20 years has really been the B cell. And, while initially there was a lot of attention paid to B cell targeted therapies in lupus in the early 2000s, as we had a better understanding of the interferon pathway, I will say that efforts into targeting that part of the pathway that resulted in anifrolumab really took precedence for a little while. But I will say in recent years, we're almost back to the future. And once again, really looking at novel ways to target those B cells and antibody production. What were some of the challenges? Well, our initial study looked at a drug called rituximab, and unfortunately, in the early and mid 2000s, there were negative trials of this medication in lupus. I will say there's lots of reasons for this. Rituximab was in fact the first biologic that was looked at in lupus, both in lupus nephritis and systemic lupus. And unfortunately, sometimes when you're first to the starting line, you're not always successful. And I will say the lupus community in general experienced a lot of challenges in terms of how we appropriately ran a clinical trial to show that a biologic agent was going to be of benefit. Collectively, there wasn't necessarily a good sense of what the right marker was of benefit. We didn't know how to control background standard of care. And in fact, most challenging is we didn't have a fundamental understanding of how to control the use of prednisone in our trials so that it didn't send a false signal in our placebo arms. And so for any number of reasons, rituximab was a negative study, although you will see in some guidelines, and in fact, there may be some of you on the call who have received this drug, we still do turn to this agent sometimes as a rescue therapy because fundamentally the manner with which it works makes a lot of sense in the pathophysiology of the disease. And we have many, case studies and registry-based studies that show that in lupus patients, this still can be an effective agent. But as of today, it is not specifically approved for the treatment of lupus in Canada. Following the rituximab failures, there were several other agents that looked at targeting the function of B cells over the next decade, which unfortunately, due to either a lack of efficacy in early studies or due to adverse events like severe infections, all fell by the wayside. And that was until belimumab or Benlysta was developed. And as I said earlier, this was the first new therapy approved for lupus management in 60 years. Now, although it's been around for the better part of a decade, initially when it came to market, we certainly did have some issues of access, and it certainly was not a therapy that was accessible for all lupus patients. And there was a lot of questions, at least initially, about who the right patients were to give this medication to, because it was, in fact, the very first biologic approved in lupus. Now, the good news here is that there's many different and novel ways for us to target B cells. And what I've shown you in this drawing here is basically the life cycle of a B cell as it grows from a very immature state of existence, even starting as a stem cell, all the way through its growth and maturation into what's called a plasma cell, which are very long-lived cells that hide out in places like the bone marrow and are responsible for the production of many of the autoantibodies that we see in lupus. And the tricky thing with our previous existing therapy is that we had a really hard time targeting some of these very mature B cells. And while we could treat and eliminate many of its family members at earlier stages of development, those plasma cells were pesky, difficult to target and difficult to get at. And rituximab, again, is the kind of first proof of concept, targeted a marker on a B cell, something called CD20. So, in the lab, we're able to identify different populations of B cells by their receptors or by markers on their cell surface. And one of the ways that we've learned to target these cells in rheumatology is by developing antibodies that are able to recognize these markers on B cells and mark them for deletion or depletion. And you can see here that CD20 there in that blue bar in the middle of your screen, while present on a number of different types of B cells, does not necessarily affect all of them. And in fact, you can see that plasmablasts and more so the plasma cell do not carry these cell surface markers. And so one of the theories between why rituximab was not nearly as effective as we thought it would be was that perhaps it just wasn't effective enough at targeting the full lineage of B cells, and therefore allowing for a repository of bad apple cells, so to speak, to exist and hide out in the body and continue to make their antibodies against parts of our lupus patients. And so we've realized that different strategies, the ability to target different markers on our B cells, might be necessary to have a more effective impact on this particular target when it comes to treating lupus. And so we're going to have a look at some of these right now. And in fact, there's a number of drugs in clinical study. So one of the other markers on B cells that we can target is something called the BAFF receptor. Now, Dr. Steinem talked a little bit to you about BAFF because that's the cytokine that Benlysta targets in lupus when it's administered. Now, BAFF floats around in the bloodstream, and belimumab is good at soaking up all of that BAFF, so it doesn't have as much of a chance to interact with cells and cause disease activity. But what would happen if we didn't just target the cytokine itself, but we also were able to target the receptor on immature and more mature B cells? And so this has led to the creation of a drug, again, that is currently in a phase 3 clinical trial called ianalumab. And this medication targets two things. It doesn't just target soluble BAFF or BLyS, which is the other name for BAFF. And you can see in the pictorial there on the left of your screen, there's belimumab, and belimumab as a medication targets that soluble BAFF and doesn't allow it to bind to cells and have its effect. But this new therapy not only tackles soluble BAFF, but in fact also binds to the receptor that BAFF interacts with in order to have its effects. And in doing so, it not only inhibits BAFF activity, but also helps to mark the B cell for depletion. And so it's a dual agent. Hopefully, two times as potentially impactful as belimumab. And again, what I'm showing you here are recently published results of a phase 2 study of this medication in the context of lupus. And what you can see here in this graph is the trajectory of patients who were initially in the first 28 weeks of the clinical trial, or the double-blinded portion where some patients got active drug and some patients got placebo. And in the blue line there, you see the number of patients that were able to achieve the primary endpoint, which again is that SRI-4 response that I talked about. And you can see at about the 28-week mark, almost 60% of patients were able to achieve that outcome, while just over 20% of patients in the placebo arm were able to have that outcome. So a fairly significant difference between the group that was on treatment versus the group that wasn't. And then what happened at week 28 is the individuals who unfortunately were randomized to get the placebo were given access to the active therapy. And what you can see over the next 22 weeks is you see those patients catch up to the cohort that were on the drug the whole time. So in the group that had the initial response, you can see that response was maintained at a high level over the course of an entire year. And in the patients who weren't given access to therapy, they caught up rapidly and were able to achieve the same outcome in about 4.5 months. And so this is a very positive signal, and again, one that we hope carries through to that phase 3 clinical trial. And if so, again, it may prove a different way for targeting B cells and their activity that could be of benefit to patients who perhaps don't respond to our other therapies. The other way that we can target B cells is by targeting a cell surface marker called CD19. And what you can see here about CD19 compared to CD20 and compared to that BAFF-R receptor is you see that this is a cell marker that really extends across the whole spectrum of B cell lineage and lifeline. And so, in theory, if we're able to target this particular cell surface marker with our therapies, we can very profoundly lead to depletion of these bad apple B cells and hopefully see improvement in lupus patients' disease activity. And this is, of course, yet another exciting target, including, as we're about to get to, one of the targets of our CAR T cell therapy. But before we get to that, I'm going to show you really briefly about another drug, again, in phase 3 clinical trials, and in this case, specifically in patients with lupus nephritis. And this is obinutuzumab. This is the Nobility study, which is a phase 2 study of this medication in patients with moderate to severe lupus nephritis. And again, like many of our medications, this drug was added on to what we consider to be standard of care and compared to a group of patients who were on standard of care but got a placebo. Now, this was a slightly longer study. Lupus nephritis studies, in order to clearly show benefit and sustainability of benefit, usually run for 2 years. So you can see here, instead of the other phase 2 studies I showed you that were only 24 or 32 weeks, this is a 104-week study. So these patients were followed for 2 years, which is a long time to run a clinical trial. And what you see here on some of these slides is one, following the treatment, which is the blue line, you see rapid improvement in many of the blood markers of lupus activity that as rheumatologists we follow. So in those top two panels, you see that complement levels statistically significantly normalized, and you see that double-stranded DNA antibodies rapidly dropped. And we know that when complement is normal and DNA levels are low, immunologically, your lupus is quieting down. So these are very important markers for us of efficacy because it shows that we're getting to the heart of really the underlying pathophysiology of the disease. And from a kidney standpoint, importantly, what we saw in this trial is that in the treatment group, we saw kidney function improve and stay improved. And we measure that by something called the eGFR, which is a measurement of how well your kidneys are basically cleaning the blood. And you can see in that blue line, the patients on treatment had a significant improvement in their kidney function that was sustained over 2 years. And we also saw a rapid and dramatic reduction in the amount of protein that the kidneys were leaking. Again, protein and too much protein leaking in the urine is again a sign of active lupus nephritis, active inflammation in the kidneys. And so improvement of protein shows that that inflammation is settled and that the kidneys are returning back to their normal state of function. So this was all very, these are all very exciting results. And what I can tell you is about a week ago, we got a news flash that the phase 3 study of this drug in lupus nephritis specifically has completed, and the data analysis has shown positive results. Now, they have not released those results yet, so I don't have specific outcome measures or markers. I would imagine those are likely going to be made aware later on this month. We have a big International Rheumatology Congress happening in November in Washington, where I would guess some of these results are going to be unveiled, but we did get a signal that the phase 3 study in lupus nephritis was positive. And with this in place, it really helps to pave the way for what hopefully will be a rapid approval by the FDA in the United States. They're usually the first ones off the mat, and then hopefully later on by Health Canada as well. I will say that there are ongoing studies of this medication in systemic lupus, much like the same population that led to the approval for anifrolumab and Benlysta. We don't have any data for this yet, so I can't make any comments there, but we're hopeful that the same degree of effectiveness that we've seen with this strategy, targeting CD19 on B cells in lupus nephritis, carries through to systemic lupus as well. So stay tuned. This, in fact, may be the next biologic that comes to market, particularly in the case of lupus nephritis. All right, and that brings us to really what is the talk of the rheumatology community. You can't go anywhere and listen to a lupus talk these days without hearing about CAR T cells. So what are these? What does CAR stand for? CAR stands for chimeric antigen receptor. And basically these are genetically engineered cell surface receptors that are encoded by DNA. that are transferred into a patient's own immune cells, in this case, T cells. And initially, this strategy, as in many cases, was not developed for rheumatology specifically. Was actually developed as a very powerful tool for managing B cell malignancies. And so there's been extensive use of these therapies in recent years in the oncology world. Why is this a particularly effective treatment? Well, unlike some of the medications that we use, and in particular the monoclonal antibodies or biologics that are very large molecules, these cells have a real natural ability to infiltrate tissues. Um, Rather than biologic medications where the levels drop off over time, they can actually expand and they can survive for long periods of time to have long-term effects on the immune system. And so because they can really get into any tissue of the body, they grow to a point, they're able to survive long periods of time, they're able to accomplish things that basically our drugs just can't. And that's getting over the issues of having, bad apple B cells, potentially escape the impact of other therapies. Um, CD19 directed CAR T cells, again, have come to rise because of as I showed you, the wide breadth of B cells in terms of the lineage that have CD19 as a marker. But what you can see from the little chart here to the right, there are many other cell surface markers that we could develop CAR T cells for that would also, more definitively target things like plasma cells and plasmablasts and memory cells, which are the longest lasting B cells, the ones that hide out the best. And we think in many cases, the existence of which are responsible for recurrent disease flares in lupus patients, because with many of our existing therapies, we just can't quite get at them. And so part of the debate right now, at least, is which one of these cell surface targets is the best strategy to go after in terms of developing these special immune cells, these special T cells to have our effect. But to date, it's the CD19 T cells that have been developed and been utilized in the clinic. And these were first used for the first time in autoimmune disease, at least rheumatologic disease, about 3 years ago in 2021. And this was in a patient who had severe lupus that was treatment refractory. So someone who had literally failed every single other therapy and continued to have very active disease, including in the kidneys. And so in that initial case report of one patient, it was found that this experimental therapy at that time was quite well tolerated. Um, it rapidly depleted the B cells, and this patient had significant improvement of their disease within 3 months of the initial treatment. So much so that over time, they were able to stop all other immunosuppressive treatment. So as I told you, in lupus, particularly in clinical trials or with these novel therapies, we're usually adding on top of rather than in place of other therapies. And in this patient's case, any of the other existing therapies ultimately were stopped because their disease responded so well. And critically, because you might ask yourself the question, well, if we're doing such a good job of taking out all these B cells, what does that mean for the long-term health of the patient? And you may say, well, we need these cells, to protect us from infection, amongst other things. And the good news is that over time, this patient's B cell population came back. So it did grow back, but even though the B cells came back, they were fundamentally different. They did not behave like autoreactive B cells anymore. And while they repopulated, there was not any disease recurrence. And so we had these new naive B cells that had formed in this patient that did not trigger disease. And hence a lot of the excitement, although just in one patient. Fundamentally, what's happening here when we use one of these therapies? Well, currently, what happens is that a patient is set up for a process called apheresis. And this is where they would present to a hospital, potentially an oncology clinic. And through the use of IV insertion and blood draws, would have essentially a population of their own T cells harvested for growth. Now, as part of that process and to set the stage for reinfusion of these medications and for them to have a fertile environment with to work, patients currently are also getting single doses of significant immunosuppression to wipe out a lot of the other potential immune cells that might cause problems. And herein lies, at least to date, one of the risks of this therapy. The first risk is, what are the potential, side effects or risks of lymphodepletion, usually done in combination with a drug called cyclophosphamide, which some of you may have heard, which is a very powerful immunosuppressive agent. After a patient's own T cells are harvested, we call that autologous, meaning it's coming from you. They're then enriched. Well, what does that mean? Effectively, they're harvested and they're expanded and they're grown so that you go from potentially a very small population of your own T cells to millions and millions that can then be used and are ready for action. Now, what are some of the risks here? Well, theoretically, in lupus, not only are the B cells a problem, but a patient may also have autoimmune or autoreactive T cells that we don't necessarily, want to be expanding and potentially reinfusing in a patient. And in some of these T cells, you may have a few duds, T cells that just aren't fit for duty. Now, hopefully that is not the majority of the millions of cells that are being produced. Now, in the lab, once you have these T cells enriched, you then are able, through gene editing or through the use of a virus, to, in a sense, infect them with a receptor that knows how to recognize the CD19 marker on B cells. And this is kind of, the genius of what we can accomplish in an immune lab these days. Effectively, we're creating our own, novel specific T cells who are only going to target one thing, and that's that cell surface marker in the body. Once you've done that editing and you now have created these CAR T cells, you expand them. So again, you have many more of them, and then you reinfuse them into the patient. And it's at this stage that they are then able to take effect, seeking out their targets. And you can see here CD19 CAR T cell and kind of the green on the right side of the page there. And because all of these different types of B cells, the pro B cells, pre B cells, very immature cells, and more mature ones at the memory and plasmablast level carry this cell surface marker, these CAR T cells can basically go and hunt these bad apple B cells down, and in some cases, not bad apple B cells, but just generally B cells, and eliminate them. So what's happening in the clinic? Well, once the drugs are infused here, you can see a very rapid decline in the autoimmune B cell population. Usually within the first 1 to 2 weeks, we see a dramatic drop in these population of cells. And while we see an initial surge of the T cells that have gone into the blood to do the hard work, over the course of about 100 days, they slowly die out as well. So these are not cells that exist in your body forever, but over a period of about 100 days, they're able to, as we said before, infiltrate tissue, identify the target cells, and eliminate them. And then after that 100th day, what do we see? Well, we see what's called a reconstitution phase, meaning existing naive B cells that had been spared deletion because they were at a very early stage and didn't have that marker start to repopulate the body with new B cells. And these are naive B cells that, as I said, do not seem to have the same autoreactivity as the old B cells that we eliminated. And what we want to see here is that not only do we see these cellular changes, but we see the disease quiet down, we see the markers of autoimmunity die down, and hopefully we see a sustained drug-free remission. What does that mean? It means that there's no other therapies being used to treat the disease and that we have a prolonged resolution of the immune attack. So this is heady stuff. So how's it borne out? Well, I can tell you that to date, there have been 8 lupus patients published in the literature who have undergone this treatment and are now at various stages of long-term follow-up, many of them, though, 24 months out from their initial treatment. So almost 2 years, which isn't a short time, but it's not a long time. what has happened to these patients? And this was data that was just recently published about 4 or 5 months ago in the New England Journal of Medicine. Well, if you look at patients 1 through 8, first of um, all of these patients have achieved what we consider to be the most important targets in terms of disease control. One, they have all achieved a lupus low disease activity score. And what that means is there's no signs of clinically active lupus. There's no major organ threatening disease, and they're on prednisone, 5 milligrams or less. But even more impressive is that each of these patients has also achieved what we call a remission state, which is an extremely stringent endpoint that I can tell you that in the clinical trial world, within 1 year, probably less than 10% of lupus patients on active biologics are able to achieve. So this is an incredibly difficult endpoint to hit. And you see that each of these patients at 2 years has achieved a remission state in their lupus. You can also see that based on the SLEDAI score, and again, the SLEDAI score is a marker of how active lupus is in various parts of the body. All of these patients at 24 months have a SLEDAI score of 0. That means the lupus is not active at all. And again, would go along with the fact that these patients are in a remission state. Again, a very difficult outcome to achieve because not only does a SLEDAI score of 0 mean that the lupus is not active clinically, it means that when we look at the immunologic markers of disease activity, your complement levels, your double-stranded DNA antibody levels, they're all normal. Next up, every single one of these patients is off steroids at 2 years. And all of these patients are off their immunosuppressive drugs at 2 years, which I can tell you, I think when we present this data to our colleagues, and even just me talking about it here, it really, it's incredible. It's almost too good to be true in a sense, but the data is the data at 24 months. And so we have a very small population of patients who are given this treatment, all very severe lupus, who 2 years later are in a remission state with no clinical or immunologic disease activity and who have achieved that being off prednisone and without any other background immunosuppressive drugs. It's pretty incredible. And then just goes to show you some of the trajectory of how quickly we saw some of these parameters proceed in these patients. And so if you look at the SLEDAI score, again, a marker of disease activity, you see following treatment, within 3 months, there was a dramatic normalization of disease activity, which correlated with complete normalization within 3 months of the double-stranded DNA levels, normalization of complement levels, and in those patients with lupus nephritis, a dramatic improvement in their protein excretion, showing improvement in the kidneys as well. So at 2 years, while there was lots of benefits, we saw the improvements after that infusion within 3 months. Now, what about safety? And you can, imagine some of the concerns that we would have at such a profound and rapid depletion of B cell populations in a person's body. And certainly there have been some infections to date. So you can see upper respiratory tract infections, and you see a number of COVID infections in some of these patients at various time points. None of these were serious infections. None of them were fatal infections and were managed with standard of care. And probably not a lot different than we might see in lupus patient populations who required steroids and immunosuppressives long-term as well. Importantly, we did not see during the infusion process any significant reactions to the infusions as well, which is another important thing to take note of. So, you might be able to hear it in my voice. This is really exciting, and hopefully exciting for all of you. But there's always a but in these things. It's early. It's very, very early. And there is still a very long road ahead before I think we become, anywhere close to comfortable to saying that this is going to be, an ongoing or long-term therapy for our lupus patients. Sorry for a bit of the cold water here. We need many more patients treated. 8 is simply not enough. In our hierarchy of evidence, what we really need are those really good phase 3 clinical trials to be developed and run, much like we've seen with some of our other therapies, with many more patients treated and longer term follow-up to know what the true impact of this therapy is going to be in a larger population of patients, what the sustainability of the therapy is going to be longer term, and what the true safety is going to be long term as well. So it's a great initial signal, but there's still a lot of work that needs to be done, to really bring this treatment to the fore in lupus patients. Um, I think we need to still answer questions about which target is the most beneficial. I mean, CD19 looks really good, but are there other potential CAR T cells with different targets that could be developed that might be even better? I will say there are a number of other CAR T cell in development that target different B cell surface cytokine markers. Who are we going to use this treatment in? The reality I will tell you is going to be that this is unlikely at any point in time to be a treatment for all lupus patients. There is significant risk involved. There is significant cost involved. I will tell you, if you thought the $21,000 that Dr. Steinman, quoted for belimumab was a lot of money, multiply that by 10 for a course of CAR T cell therapy currently today in this day and age. It's not to say it's always going to be that expensive. It's not to say there may not be other ways to utilize this therapy, but it just highlights that we are going to need to be selective about who receives this treatment in the lupus world. The other issue is one of access. Right now, to administer this treatment requires the use of a lot of hospital resources. And I will say right now, while this strategy is used, fairly, I won't know if it's commonly, but is used often in by our hematology oncology colleagues. The demand for this treatment in oncology right now, at least locally, outstrips our ability to deliver it. And I will say, in conversations with some of my oncology colleagues in Hamilton, there are some patients who, in fact, have to be sent to the United States to receive CAR T cell therapy because we simply don't have enough resources right now in order to treat oncology patients where this is a proven therapy. And so one of the other things we're going to have to grapple with is access to resources. And how are we going to potentially administer this therapy to patients in an equitable and timely fashion if, in fact, long-term it does prove to be successful. So some final thoughts for me as we come to time. I apologize, I've run a bit over. As hopefully I've imparted on you, we've just made such enormous strides just in the last decade. Dr. Steinman talked about the lupus renaissance. I can tell you there's just so much activity happening in the research world right now. There's lots of excitement. We've got more lupus-specific targeted therapies. We've managed to improve access to the existing targeted therapies that we have, and increasingly we're understanding better targets for disease control and using less steroids to achieve our benefits. We are very close to that next wave of therapies. We're not going to be waiting another 10 to 20 years in between lupus treatments anymore. Some of these may target interferon pathways. Some of them are going to continue to target the B cell pathways. And then, of course, we come to the CAR T cell therapy. Is this hope for life without lupus? It's an exceptionally compelling story. There's lots of ifs. There's lots of uncertainty, but it's going to be very exciting to watch this unfold over the coming years. And with that, I will end my talk, and I'm happy to take questions. Thank you.
Thank you so much, Dr. Matsos. That was so fascinating. A lot of information to take in, but I really appreciated the path you took us through to really understanding the cells leading up to this and all the various treatments. I do have a bunch of questions from someone, but there's a couple of comments I wanted to make. One of the things that I thought was lovely that you commented on was trials that fail. While they're very disappointing, I'm really appreciative that they then help inform new research and innovative ideas. And so if anybody's, paying attention to news reports about these kinds of trials and then see that there's a failure, don't be discouraged, because really smart scientists are then building upon that for what's leading to these great things. Um, I also love that some drugs used for other rheumatic diseases are being tested used for lupus. And this is another sort of an advocacy point for lupus patients to try to get into rheumatologists who focus on lupus, because they're the ones that really you can appreciate the care from them, to know about using other options such as rituximab or those kinds of things as well. Is really good. I was wondering about the JAK-STAT inhibitor. Should lupus patients specifically ask the rheumatologists about using any of those treatments?
Yeah, Lisa, it's a good question. I think it's too early. Certainly it would be considered an off-label use of those therapies. They have not yet been proven in clinical trials. So there's not a ton of evidence behind them yet, where I think a significant number of rheumatologists might feel comfortable deploying them outside of where they're approved by Health Canada. So I think we have to wait and see what the data shows.
Gotcha. Okay, that's great. Um, um, Joe has asked, how much do you think advocacy has played a role in breaking the 60-year gap in new treatments? For example, was it lupus was relatively unknown and not typically researched, or was it more a matter of waiting for these drugs to be designed before they could be researched in lupus patients specifically?
Yeah, Joe, that's a great question. I mean, advocacy, I will say particularly in the last 20 years has just been massive. Both, really across the world, especially in North America, as patient groups like Lupus Ontario sort of clamored for better treatments, and better outcomes. And that's really driven the field forward. I think to some degree we had to wait until we had a better fundamental understanding of what was driving disease activity in a lot of patients. And the reality is that lupus is just such a complex disease. I mean, many of the illnesses that as rheumatologists we treat are complex, but lupus is just next level, and it's such a heterogeneous disease, as I'm sure you all are well aware, that it really took us a long time to find some of these commonalities in the pathophysiology that allowed us, initially at least, to say, okay, well, we know this. What do we have readily available to try? And so the idea of B cell depletion with rituximab, which again was an oncology treatment and an RA treatment first before we tried it in lupus. And then more specifically, we found targets that made sense in lupus patient populations. So, BLIS or BAFF, which is the cytokine that Benlysta or belimumab targets, was found in research to be found at much higher levels in lupus patients compared to patients with other autoimmune diseases like rheumatoid arthritis, and much higher than in healthy populations. So obviously stood out as an obvious target that allowed for drug development meant for a lupus patient. I mean, belimumab is not really approved to use in other illnesses right now outside of lupus. It is a lupus specific therapy, and similarly with anifrolumab. We know that in lupus patients, they have something called a high interferon gene signature, meaning that the genes that are activated by the cytokine family that are the type 1 interferons are activated at a high rate, again, compared to other autoimmune diseases like RA and compared to healthy patient populations. And we have a much more fundamental understanding of some of the genetic risk factors as well as lupus, to know that there's genetic polymorphisms or abnormalities that happen in the genes of lupus patients that pertains to how the interferons function in the body, that makes it, a key player in the disease. So again, understanding that, really led to the targeting of interferon as a pathway in these molecules. And to Lisa's point, our first attempts at targeting interferon was not anifrolumab. It was actually a whole other family of antibodies, monoclonal antibodies that tried to, soak up as much of the interferon cytokines in the blood as possible. And I will tell you that those trials failed miserably. Again, even though, the pathway made a lot of sense in terms of the pathophysiology. And it wasn't until we tried to tackle interferon a completely different way by blocking a receptor with anifrolumab rather than all the cytokines themselves that we got success. So even sometimes when we know a target makes sense, It's finding the right way to tackle that target with our therapies that we struggle with, and why you're seeing that there's a number of different drugs that are all kind of trying to interfere with the same pathway. We're, just trying different patterns of attack to see what works best.
Mhm. That's really great. Joe also asked something about what happens sort of in studies. So we said in the initial CD19 CAR T therapy trials with the severely refractory patients, what happened to them after the study? Were they able to continue using the therapy, or did they have to return to more established treatments that
Yeah,
had failed?
that's a great question. Yeah, so most studies now, not all, but most studies have something called a long-term extension. And so, when we talk to patients, about participating in clinical trial research, which is, a conversation I have fairly frequently with the patients in our clinic, there has to be an acceptance that when you decide to partake in clinical trial, there's certainly a chance that you might get early access to a therapy that could be beneficial, but we don't know that. And we have some certainty as to the safety data, but not complete certainty. But there's also a chance that, for a period of that trial, you're going to get nothing besides the usual standard of care, an excellent clinical follow-up. I mean, patients in clinical trials, we see them every 3 to 4 weeks. So you're very closely monitored, which certainly helps with outcomes as well. But the carrot, so to speak, so, a patient may say, well, look, I don't like the odds, of me being, putting all this effort to join a clinical trial and being a guinea pig, so to speak, if, I'm going to be given sugar water or a fake drug for all my trouble. And so the carrot, so to speak, for patients who are so inclined to participate is that in some studies during the initial trial period, they get switched over to drug, like what happened in the one study that I presented. Or in some other studies, if they can make it through the end of the full clinical trial, they're then entered into this open label extension, meaning that we know that they're going to get the drug at that point for sure. And then we enter into a different phase, which is more of a monitoring phase. So, it's less experimental because as an investigator, we potentially biased because we know you're actually getting the drug now. But again, potentially gives lupus patients access to some of these therapies that may be approved sometimes 5 years before they've actually hit the market. And many sponsors will continue patients on the active drug during that time because they're also still gaining information. They're gaining information about, well, what's the long-term success of this drug? What are the safety signals? So a drug like anifrolumab that's only been on the market for about 2 to 3 years. In fact, there have been some patients on this drug for well over 5 to 6 years, even longer because they were part of the phase 3 clinical trials and then went into the long-term extension. And so we've been able to gain, even before market, extensive experience from these patients who have made the effort to participate and help us to generate data and understanding of the therapies.
Mm. Thank you so much. This is another reason why I'm going to encourage any lupus warriors to participate in trials if you're given the opportunity, because you are helping further the science for all of us, which is lovely, and hopefully get amazing care for yourself as well as you try new things. Just bear with us, people, if you want to hang in. We just have a couple more questions as we're going to wrap up. So I'm going to say this word wrong, but will obin
Obinutuzumab.
yeah, thanks. Be for a certain stage or severity of lupus nephritis or anyone with the diagnosis?
That's a great question. So the clinical trial for which we were told there's positive phase 3 results. Again, I don't know the specifics of those results. Besides the news release we all saw that said that it was a success, was specifically for patients with the most severe form of lupus nephritis, so class 3 and class 4, which typically for those lupus patients who suffer with this, unfortunately, carries a lot of the risk of long-term morbidity and long-term kidney damage. So initially, again, depending on how the results look and depending on what the regulatory bodies think of those results. If in fact it is approved, initially it looks like it would just be in those patients with severe lupus nephritis. What we're hoping is that the other study that's sort of being piggybacked, where this drug is being studied in lupus patients, but without severe kidney disease, other manifestations, we're hopeful to see similar results in that patient population in the clinical trials, but we're at a much earlier stage of study. So we don't have a lot of data there yet, but we certainly hope that ends up being a positive trial. And if so, may open up the use of this therapy in all forms of lupus, not just severe lupus nephritis.
Amazing. I love when something is for one area and then can steamroll into all the other areas for us as well. When you were talking about how the CAR T cell therapy actually works and what it does, it just made me totally think that we are having these little mini assassins come into our body to take care of stuff and take care of the bad stuff. So it's great. I'm looking forward to these opportunities. Do you know how long it approximately takes to grow the modified cells before
It's not long. I believe it's only a week or two that it takes in between infusions. So this is not a long drawn out process in order to multiply the cells for use.
Okay. And we know this still quite a ways. But if in theory the CAR T cell therapy cells now are, lowering your immunity quite low, well, it targets all these things. Can one assume that it would also then help reduce, eliminate, control other autoimmune diseases that you would have, especially as lupus patients have so many comorbidities?
Yeah, so I mean, I didn't show you the full. So there were actually 15 patients in that cohort that were treated with CAR T cell therapy. 8 of them were lupus patients, but there were also patients with myositis, and there were also patients with systemic sclerosis or scleroderma who were also involved in that cohort as well. And I can't speak specifically to the data on those patients beyond saying that there was also improvement in those clinical states as well. So, really CAR T cell therapy, theoretically speaking, could be utilized in any autoimmune disease, at least the CD19 ones, where the B cell is felt to play, a central role in the pathophysiology of the disease. That's the
Mm. That's amazing. Like, speaking from somebody who has 4 autoimmune diseases, one of which is Sjogren's and the other lupus, like they're so common for the symptoms and the way it presents that, I would think, hey, if this is working for one, it probably would definitely help the other. So yeah, that'd be exciting to see. So I'm just gonna finish with one last question is we know it's too early to say for certain, but if CAR T does turn out to be a one-time treatment, and just I know you were saying long-term remission and weren't saying cure.
hope.
Really lovely to see if it was cure, but I love that you're using that term. But so say it is leading to long-term remission. Would highlighting the cost savings of reducing, your future hospital admissions, the lupus management, all that stuff. We already know that's an advocacy effort we're already trying to leverage for biologics, because let's be clear, if biologics are helping you, you're not going to the ER all the time, you're not maybe on dialysis, you're getting back into the workforce, contributing to the economy. So we're already working on those types of advocacy stuff for that. But do you think that that might end up being a pathway for advocacy and help in the future if this would offset all of those other
Yeah,
costs?
at least that's certainly a great point and important consideration. I will say that I think as this technique becomes more adopted again, depending on how these patients do and safety and all that, you're going to see more and more of these therapies coming to market, which will likely help to bring down the costs. The other thing that's being looked at is this concept of off-the-shelf CAR T cells, meaning that through the use of donors whose T cells are harvested and then matched to the individual recipient who needs them, we're actually able to bring the costs down because we're eliminating the need for the whole collection phase and also like that in a patient. So, which may mean that these therapies could potentially, one day, and this is all sort of pie in the sky right now, one day be transfused outside of hospital settings, which again would bring down the resource requirement and bring down the total cost of therapy. So, there's multiple, sort of potential ways that over time, if this bears out, that the cost may come down up to a point, who knows how much, where it's easier to access, more widely available, and less resource intensive. But your initial point that, a one-time upfront treatment that leads to disease remission for a long time, while reducing, the long-term comorbidity that comes with active disease and the use of these immunosuppressive therapies, is really a win-win, and hopefully regulators can see it that way as well.
Mm. Yeah, another reason why groups like Lupus Ontario and others are really important for advocacy to work alongside scientists and doctors as well. So thank you so much, Dr. Matsos, for your really informative information. I'm so excited to see what's going to happen in the years to come. Even five years from now, I think this conversation is going to be different and expansive and even beyond that. So we really appreciate you laying it out in a way that can make sense to us.
Thanks
So
again, Lisa, for the invitation.
absolutely. Thanks everybody for joining us and hanging in for this. Again, if you haven't seen the other sessions, please watch our YouTube channel for the other videos. If you have any questions about anything you've seen or comments, please visit us at lupusontario.org. And otherwise, we wish you a great night.